Good morning, everyone. So, dear colleagues and friends, welcome to our new ELSI Dialogues webinar. My name is Ilaria Colussi, and I am Data Protection Specialist for BBMRI ERIC. I will be moderating this session. Today, we are going to be exploring the content of the Nagoya Protocol, which is an international agreement aiming at sharing the benefits arising from the utilization of genetic resources in a fair and equitable way. It is a supplementary agreement to the 1992 Convention on Biological Diversity, being in force since 2014. The Nagoya Protocol does not explicitly cover human genetic material, and if research is done on human DNA, RNA, and derivatives, this is out of the scope of Nagoya Protocol. However, when research focuses on materials associated with humans, such as pathogens and microbiota or microorganisms, this research might be in scope. Thus, when a biobank, in addition to its usual activities of receiving, processing, storing, and distributing samples, performs a sequencing of human pathogens, the Nagoya Protocol might be applied, impacting how biological materials are accessed and exchanged. For this reason, navigating the protocol's provisions with confidence can be complex for researchers, biobanks, and institutions. For helping us clarify how Nagoya Protocol applies to biobanks and its challenges, we have today the honor of hosting three excellent speakers who will provide us with practical knowledge, expert perspectives, and actionable strategies. But before we start, I will just give you a brief background about BBMRI ERIC, which stands for the Biobanking and Biomolecular Resources Research Infrastructure, European Research Infrastructure Consortium. It has the scope to facilitate access to sample and data collections across Europe to advance biomedical research for the benefit of the European citizens. To reach this goal, BBMRI ERIC relies on a network of national nodes in the member countries, as well as a team of interdisciplinary experts from medicine, social science, ethics, law, IT, quality management, patient communities, and industry partners. BBMRI ERIC ELSI Services and Research, named ELSI, provides support on ethical, legal, and societal issues, operating in partnership with a network of experts from academia and practice from across both our national nodes and project partners, alongside three key cornerstones: research, service, and training. This webinar is possible thanks to funding from the European Union's Horizon Europe Research and Innovation Programme. Specifically, it is supported by the CanServe project that focuses on providing cutting-edge cancer research services across Europe. For details, visit the website canserve.eu. Moreover, this webinar is also in close alignment with BBMRI ERIC's work program activities for 2025-2027, namely Strategic Objective 1, optimize the interconnection of human, animal, and environmental health research, Task 1.3, about interconnections with viral, microbial, environmental, animal, and/or plant collections. So without further delay, let's now proceed on our webinar today. The first speaker is Melania Munoz Garcia, a Costa Rican biologist working in the Science Policy Department of the Leibniz Institute, the SMZ, German Collection of Microorganisms and Cell Cultures, where she serves as a project officer for the German Nagoya Protocol Hub project. She helps researchers navigate their obligations under the Nagoya Protocol and the EU regulation on ABS compliance. Additionally, Melania is an active member of the Digital Sequence Information Scientific Network and participates in international negotiations on benefit sharing from the use of digital sequence information. Melania will introduce us to the Nagoya Protocol, its application to biobanks, and the implementation across Europe. So, Melania, the floor is all yours. Thank you.
Then let's start talking about the Nagoya Protocol and its importance and relevance for biobanks. Today, we will cover the basics of the Nagoya Protocol, the relevance for biobanks and research, and the obligations under the European Union Access and Benefit Sharing Regulation, which are our obligations as scientists in the EU. First, the basics of the Nagoya Protocol. It's not a secret for anybody that the biodiversity is decreasing in all continents. And this implies a lot of important issues for the day-to-day life of human beings. Then, in 1992, the UN Convention of Biological Diversity was adopted in order to face this problem, the biodiversity loss. And it involves three main objectives: the conservation of biological diversity, the sustainable use of the biodiversity, and benefit sharing. And to implement this last objective, the Nagoya Protocol was adopted. The Nagoya Protocol is specifically on access to genetic resources and the fair and equitable sharing of benefits arising from their utilization. It was adopted in 12th of October 2014, and it is the comprehensive international framework for ABS, access and benefit sharing, and compliance. And we all should take care of it. But what is ABS about? ABS comes from access and benefit sharing, and access means using this biological material for research and development, but asking permission first. This permission should be asked specifically to governmental institutions in the country where we are taking these biological materials. And benefit sharing means giving something back, that in the basis of the non-commercial research, it could be many things, from sharing the research results, joint publications, some training, capacity building, in-kind support to scientists and communities and the government in the provider country. And also the main idea of all of this is that access and benefit sharing should contribute to conservation and sustainable use of biodiversity in the country where we are taking these samples. That from now we will call it the provider country. Which are the three main pillars of the Nagoya Protocol? So first is access. Each country can decide if they establish access rules, I mean, the laws and procedures to access the genetic resources within that country. And access is not implemented at the EU level. That means that each member state has the right to decide if they regulate access and how they regulate access. So for example, Germany and the Netherlands and other countries have already decided to not regulate access to the genetic resources. So genetic resources from Germany don't need an ABS permit. But other countries within the European Union, like Spain or France and Greece, have decided to regulate. So it is important that we are aware of these legislations and follow them. The second pillar is benefit sharing, and this is which benefits are we willing to share with the provider country. And this is most of the time subject to a contractual agreement between the provider country and the user. And the third pillar is compliance. And this means that every country should check and control that all users of genetic resources within their jurisdiction are following the access and benefit sharing rules from the provider countries. And this is regulated at the EU level with the European Union ABS Regulation 511 from 2014. And it established due diligence obligations for all users of genetic resources within the European Union. And we will study this regulation in few slides. So the Nagoya Protocol at the end is relevant for all the people or researchers that do research with the genetic resources or derivatives. Then we have to start understanding what is a genetic resources and what is a derivative. So genetic resources are all the biological materials that contain DNA or RNA. It doesn't matter if they are alive organisms, for example, in the field, or in ex situ collections like in vitro culture microorganisms, or if they are already dead. For example, if you obtain dry plants from an herbarium, it also counts as genetic resources, or for archaeological excavations and so on. So genetic resource are any material of plant, animal, or microbial or other non-human origin containing functional units of heritage. As mentioned in the introduction, the human DNA, RNA, and derivatives are excluded from the Nagoya Protocol, but pathogens and all microorganisms living on or inside human bodies are part of the scope of the Nagoya Protocol. So you must take care of that. Another exception is the plant genetic resources under the International Treaty for Plant Genetic Resources for Food and Agriculture. And this treaty has an annex 1, that include all the crops that are within the scope of the plant treaties and out of the scope of the Nagoya Protocol. And the third exclusion is the influenza strains under the Pandemic Influenza Preparedness Framework, the PIP framework. And it is important to mention that to be out of the scope of the Nagoya Protocol, the biological material and the use must be under the scope of these two agreements, the plant treaty and the PIP framework. For example, rice is within the annex of the plant treaty. If you use it for food and agriculture, then it's excluded from the Nagoya Protocol. But if you use rice to produce a cosmetic or a medicine, then it is out of the scope of the plant treaty because it's not food and agriculture and is within the scope of the Nagoya Protocol. So it is important to make this distinction. Then about derivatives. Derivatives is a word that scientists are not used to, but it is defined in the convention and the Nagoya Protocol is naturally occurring biochemical compounds resulting from the genetic expression and metabolism, even though they do not contain units of heritage. And it could include proteins, including enzymes, lipids, organic compounds, essential oils or resins or milk or honey or others, wax, for example, and other products of metabolism. So it is important to consider that it's not just DNA and RNA, but also all these biochemical compounds included in the Nagoya Protocol. And why is the Nagoya Protocol important? At the end of the day, the access and benefit sharing measures ensure that research results support the provider countries. How? Making informed decisions on conservation, sustainable use, and management of their own biodiversity. So basic research can give information to take these decisions, to make these decisions, implement innovative solutions for productive activities, for example, agriculture, fisheries, energy production, and so on, and then contribute to national sustainable development at the national level. Then also the Nagoya Protocol can contribute to build capacities for local researchers to solve local problems in the provider countries, and it can also include other monetary and non-monetary benefits that you can directly agree with the party. And what is the relevance for biobanks and research? First, let's start talking about the Leibniz Institute DSM-Z, the German collection of microorganisms and cell cultures. This is a house for the most biological diverse microbial collection worldwide. At DSM-Z, we had around 84,000 bioresources, and we received deposits, like around 2,500 deposits per year, and we distribute these strains for all around the world. We distribute around 43,000 bioresources per year. So as an institute to store, preserve, and distribute biological material, we need to fulfill international obligations for exchanging this material. And it is important to mention that we have this obligation even though the mere storage of the biological material is without conducting any research is out of the scope of the EU ABS regulation and the Nagoya Protocol. But in general, the EU ABS regulation also gives the opportunity to register the collections, and the DSM-Z is the first EU registered collection under the EU regulation. That means that everybody that would like to deposit a microorganism in the collection should also include the ABS documents, the ABS permits. And then we include all these ABS permits and documentation into our catalog. So every sample or every strain you would like to obtain from DSM-Z has the Nagoya Protocol restrictions, and you can see here in the bottom. So if the strain doesn't have any restriction, you will know about it. But if the strain has some kind of restriction, for example, this one, the catalog will describe what kind of restrictions and what kind of ABS conditions you have to follow. This was a very important effort for DSM-Z. It took 2.5 years to finish this process and include all the Nagoya Protocol documentation within the catalog, but it had very important outcomes because users can now easily fulfill their Nagoya Protocol due diligence requirements in the European Union. They can, it increases the usability and legal certainty of DSM-Z collection, and also gave a rise to the Science Policy and Internationalization Department because we gain a lot of experience related to the Nagoya Protocol and its implementation. Then in general, for ex situ collections, it is a good practice to compile and transfer all the Nagoya Protocol relevant information. It is a good practice because it facilitates the future transfer and utilization of these materials. And it is important to highlight that it's very difficult for the end user to obtain all this Nagoya Protocol relevant information if the collection do not facilitate it. So it is a lot of help for researchers to obtain this information directly from the collections from where they are obtaining the biological material. And one important thing to consider for microorganisms is the relationship between the Nagoya Protocol and the International Code of Nomenclature for Prokaryotes, the ICNP, because the ICNP requires that for new species descriptions, the scientists must deposit it into collections into different countries without restrictions. So this type strain can be transferable to third parties without these restrictions. And it is important to check all the ABS permits to see if they include restrictions, because if the permit includes restrictions, for example, do not transfer to third parties, or third parties need a permit before using this strain, the collection may not accept your deposit. So this is very important because this could also create difficulties to describe new species of microorganisms. Then the ABS principle is easy to understand. It's just asking permission to access the genetic resources and give some benefits back. But it can get a bit complicated. And I would like to explain that the ABS world has two main parts, and researchers need to understand both. So here in this infographic that you can access from the German Nagoya Protocol Hub website, explains the two phases of the ABS work. The first part is related to the obligations according to the ABS rules in provider countries. And we included key questions to identify if your research is within the scope of the national ABS. And then once you obtain your research, you can start your research, but you may also have obligations under the EU ABS regulation if you perform your research in the European Union. And why it can get complicated? Because every country has the right to decide how they do ABS, and they do ABS different. So when projects need to obtain and study samples from different countries, they have to go through the ABS process in multiple countries at the same time. So it can get complicated. And let's now talk about the obligations under the EU ABS regulation. Whether you have compliance obligations in the EU is determined by this regulation 511 from 2014. And here we have prepared some questions to help you to know if you are within the scope of the EU regulation. The first question is if your material contains DNA, RNA, and derivatives. So we already explained this, it's not that complicated. Then the tricky part is to know if you are utilizing genetic resources in the terms of the EU ABS regulation. If you are doing research on the genetic and biochemical composition of the material. So for example, the mere storing of genetic resources is not utilization. Just comparison, for example, to identify the taxonomic groups of the material is not considered utilization. And for example, just do like morphological characterization is not utilization. You must do some kind of research related to gene expression, gene function, and so on to be in the scope of utilization under the EU. Then you have to know if the material was originally obtained on or after 12th of October 2014, when the Nagoya Protocol was entered into force, and the same date as the EU regulation entered into force. And this is important for biobanks and collections because it's not the date where it was deposited in the collection, it was the date when it was originally collected in the field. It's important to make the difference. And then is the provided country party to the Nagoya Protocol, and ABS measures were in place at the time of access. This is also the fourth question you have to answer. You can find this information in the ABS clearinghouse, but when the ABS clearinghouse information is not complete, we always recommend to ask these questions or send these inquiries to the national authorities and be completely sure that the party or the country don't have ABS regulations. Then if you answer yes to all these questions, you are within the scope of the EU regulation. And there are two important things to consider: that the national ABS legislation may apply to you even if the EU regulation does not. And the EU ABS regulation scope is narrower then the scopes of the national legislations of parties of the Nagoya Protocol could be bigger, so it is important to make the difference. And even though some countries that are not party to the Nagoya Protocol, they do regulate access to genetic resources. So you don't have obligations under the EU, but you do have obligations under the national laws. Then the obligations, what are the obligations under the EU ABS regulation? Well, what the national authorities will check if you comply with the ABS measures in the provider country. For this, you need to seek, keep, and transfer ABS information to subsequent users. So seek information is obtaining all the permits you need, keep it for 20 years after the end of the utilization, and transfer it to all third parties. And after that, you need to exercise the due diligence declaration. It's pretty easy. You go to the Declare portal. There is an EU portal, and you declare that you obtained your samples legally, and you upload your permits. Then you have to use the material according to your permit, and if you don't have sufficient information, you have to discontinue utilization to avoid fines. And it is also important for transferring material. So it is important to highlight that material transfer agreements, the MTIs that we are used to, are not sufficient to fulfill ABS information obligations. The MTAs are usually documents that are signed between two different institutions, and the ABS permits are signed or granted by the authorities of the country. So this is the main difference. And for transferring, the ones sharing the material have to transfer the Nagoya Protocol relevant information, and the ones receiving the material and utilizing the materials are the ones that need to comply with the ABS obligations. And here we include a list of questions that could be useful for you in order to be sure that all documentation you are receiving is okay. You need to check if you're receiving all documentation and if you are enabled to do your research with that documentation, or if you need additional permits. And you need to check also what kind of restrictions are attached to that biological material coming from the Nagoya Protocol permits. And the other thing that's happening in the EU is the compliance checks that are performed by the authorities in each of the member states. In the case of Germany, the German Federal Agency for Nature Conservation is the one implementing these user checks, and they are checking all the institutions in Germany to see if they comply with the EU regulation. Then it includes answer a questionnaire, provide copies of all the documents and the Nagoya Protocol permits and so on. Sometimes it's necessary to allow access to your premises, and sometimes even though provide some samples. And it's an obligation under the EU regulation to support these user checks. And after the checks, there are implications of non-compliance. So if you have to take it seriously because there are important implications. For example, in Germany, fines could be up to 50,000 euros. They can order to stop your research, they can confiscate your material, and then can do publication embargoes or retractions. And then you can also be sanctioned abroad from the provider country's legislation that includes from fines and also jail terms in some countries. So yes, you could be sanctioned twice, in Germany and from the provider country. And there are some other non-regulatory consequences like loss of reputation, loss of trust in provider countries, and be included in blacklists. And here we can see some examples of important scientific journals that are exposing researchers that haven't complied with the Nagoya Protocol. So it is serious. But we have this project and everything could be a bit complicated, but we are here to help. I invite you to visit the Nagoya Protocol Hub project. We do this kind of information sessions. We have a help desk, and we also produce easy to understand material related to the Nagoya Protocol that you can check and use. And with this, I will finish my presentation. Thank you very much for your attention.
Thanks a lot, Melania. It was a great presentation, and you perfectly brought us into the topic of Nagoya Protocol, especially showing the impact of this to biobanks in Europe, and also considering the EU regulation at stake and national law, and how these three levels intersect to each other, which is legally very interesting and also very challenging, I would say. So extremely helpful for our community. Thanks a lot. The next speaker is Josephine Ultry, who is a Master of Science trained life sciences and technology engineer from the Swiss Federal Institute of Technology in Lausanne, with extensive experience in quality management across the biopharmaceutical, medical, and academic sectors. Currently, she is appointed as Quality Officer at Swiss Biobanking Platform, the reference research infrastructure for human and non-human biobanks funded by the Swiss National Science Foundation. She is also the Swiss representative in our ELSI Help Desk network and has a strong interest in the One Health approach to address the complex challenges impacting human, animal, and environmental health. Josephine will present us the Swiss context with regards to the implementation of Nagoya Protocol. So, Josephine, the floor is all yours. Please.
Okay, so hello everybody. Thanks, Ilaria, for the kind introduction. I will provide you with a brief overview on how the Nagoya Protocol has been implemented in Switzerland, and I will propose you some recommendation on how biobank can proceed to comply effectively. So I will first explain to you how the Convention on Biological Diversity and the Nagoya Protocol were translated into Swiss law. And I will present a guideline that we developed at the Swiss Biobanking Platform with a focus on three main aspects: the access to non-human genetic resources, the due diligence requirement, and finally, the benefit sharing, which is the core objective of the Nagoya Protocol. So what about Switzerland? Switzerland ratified the Nagoya Protocol in 2014, and it was included into two legal documents, the Federal Act on the Protection of Nature and Cultural Heritage, NCHA, and the Ordinance on Access to Genetic Resources and the Fair and Equitable Sharing of Benefits arising from their utilization, the NAGO Ordinance. So if there is one thing to remember from my presentation, this is that Switzerland requires due diligence for the utilization of non-human genetic resources, and also a notification before the commercialization or market approval of a product. This is the Federal Office for the Environment, the FOEN, that is responsible to check the application of the NAGO Ordinance in Switzerland. So if you go on the Access and Benefit Sharing Clearinghouse website, so you can find the link provided on my slide. You can find all the countries that ratified the protocol and also the contact information of the national focal point. So if we get specific questions from biobankers regarding a specific application of the Nagoya Protocol, we always redirect these biobankers to the national focal point. So the Nago Ordinance and the Nagoya Protocol regulate the utilization of the non-human genetic resources, but not the collection and the storage by themselves. So sometimes it can be hard to determine whether a specific biobanking activity is a utilization in the sense of the protocol. Also because essentially the precise use of the samples stored in the biobanks is most of the time not known in advance. Actually, there are several years between the time of the collection of the samples and the use of the sample for research. It's important to note that even if some biobankers are only transferring material, so they are not users in the sense of the protocol, they might still be subject to contractual obligation, like fulfilling the due diligence requirements, so meaning collecting a specific set of information and passing on this information to the researchers that will use the samples. So at the Swiss Biobanking Platform, we chose a pragmatic approach and with our own interpretation of the legal text, and we proposed a really practical application targeted for biobanks. So here's the general flow that we propose to biobanks with provider country, number 3, giving access to non-human genetic resources to a biobank in Switzerland, number 4, that will store these samples for a certain amount of time before a user, typically a researcher, will use this material for a commercial or non-commercial use, number 6, and then benefit sharing might apply between the user and the provider of these genetic resources. So I will now focus on the access to genetic resources. So if you're a Swiss biobank and you want to access non-human genetic resources from abroad, there are basically 2 cases to distinguish. First one is when the country signed the Nagoya Protocol. In this case, the rule of thumb is to check the access and benefit sharing clearinghouse website, the one that I showed before, because depending on the country, there are several requirements that might apply. The due diligence requirement, meaning collecting a specific set of information. I will come back to that later on. But also signing prior informed consent, PIC, or signing mutually agreed terms. Concerning mutually agreed terms, we refer the biobankers to a toolbox that was developed by the Swiss Academy of Science that is available on their website that includes a template for this kind of agreement. If the material comes from Switzerland, only the due diligence requirement, meaning collecting a specific set of information, is required. If the country didn't sign the Nagoya Protocol, then there is no legal requirements. However, collecting specific information, signing prior informed consent, and mutually agreed terms, this is always considered as best practice. Second topic that I wanted to focus on is the due diligence for biobanks. So in practical terms, fulfilling the due diligence requirements means collecting a specific set of information, registering this information, and passing on this information to subsequent users. The information related to the due diligence can be notified on a voluntary basis to the Federal Office for the Environment with forms that are available on their website. So we advise the biobank to record this information as a retention type to keep it for 10 years at least after the end of utilization of the samples for research, or at least as long as the sample is stored in the biobank. We also provide the biobanks with a data set for their biobank information management system, including all the information requested to fulfill the due diligence requirement. So I copied the kind of variables that were included in this data set. So as you can see, this is not rocket biobanking in the sense that it's not complicated to understand which kind of information is needed. But still, if you haven't recorded this information from the beginning, it can be hard to come back to it afterwards. For example, regarding the access date to genetic resources. Last topic that I wanted to focus on is the benefit sharing between the user and the provider, which is the main objective of the Nagoya Protocol. So according to the Nagoya Protocol, benefit sharing should apply to both commercial and non-commercial use, and it can be monetary, but it can also be non-monetary. So in Switzerland, in the Nagoya Ordinance, the Federal Office of the Environment, I'm here citing the law, encourages users to voluntarily share the benefits arising from the utilization of genetic resources in a fair and equitable way, even when there is no legal obligation to do so. So in our guideline, we proposed some example of monetary benefits like royalty payments, joint ownership of IP rights, and as non-monetary, there is research and development knowledge, training and education, and transfer of technology. Of course, they can be negotiated between Switzerland and the provider country in the mutually agreed terms and should be done in advance. So that's the end. Thank you for listening, and thanks to all the people who contributed to our guideline, in particular Francisca Bossart from the Federal Office for the Environment with the national focal point. And you can find my contact information if you have specific questions regarding my presentation.
Thanks a lot. Thanks, Josephine. It was very, very interesting as well, and you offer relevant insights on the national level, which is really important for the implementation of Nagoya Protocol. The final speaker we have is Dr. Christian de Guthrie, Scientific Project Manager at SIB, Swiss Institute of Bioinformatics. And he holds a PhD in applied genomics and co-founded a startup specializing in genomics and cryopreservation, where he developed a strong interest in metadata and long-term sample preservation. For the past 2 years, he also served as the scientific project manager for Biodiversity Genomics Europe, ensuring ethical compliance with the collection, shipment, and storage of each specimen. Christian will share his practical experience of the Nagoya Protocol application as a project manager in Swiss and pan-European projects. So, Christian, the floor is all yours. Please.
Good morning, everyone. I'm here today to talk about how the Nagoya Protocol is of interest for biobanks in the context of non-human samples. And my insights come from overseeing the compliance and the project coordination for one stream of BGE. Okay, what is BGE? BGE is Biodiversity Genomics Europe, and it is an EU-funded project dedicated to the sequencing of non-human species across Europe, including animal, plants, and fungi, but not only. And clearly, we must adhere to the Nagoya Protocol, and this influences how we store samples in biobanks, how we track metadata, and how we share the data that we produce. First of all, I would make a huge distinction between human and non-human samples. So human samples are governed, and the data that derive from the human samples are governed by GDPR, ethics committees, consent requirements, and tight restriction on data sharing for clear reasons. But for non-human samples, open data is mostly promoted to maximize the scientific benefit. However, the Nagoya protocols remain the key for us. It guarantees that countries of origin have a say in how the genetic resources are used. You can see it as the respect we show for human participants in medical research. Okay, let's try to understand what we do in a general way. So this is our workflow. And the Nagoya Protocol is at the beginning under the plan and permit section, and it will influence, as I said before, all the downstream steps. We can summarize everything in 3 sections. So we can start from the sampling and planning, and then from the sampling, and then the processing of the sample to generate the genomic sequences. And as you can see here, biobanking is a requirement in our project to guarantee the traceability and empower the future of research. And maintaining proper documentation along all the pipeline supports this traceability and compliance, reflecting some of the principles that guide human biobanking. Okay, BGE operates in more than 20 European countries. And what is important to recognize is that while the Nagoya Protocol set a broad international framework, as Melania said before, each country can interpret and implement it differently in the form of bilateral agreements. So for instance, some require additional benefit sharing agreements, while others have local biodiversity laws that go beyond the Nagoya Protocol. And it's key to understand that the Nagoya Protocol, it's one piece of the puzzle to access the genetic resources. This includes, the puzzle includes local, national, and international permitting. And we must be transparent as a European-funded project regarding the permits collected for each species that we sequence. I was asked to give this talk and to highlight some of the challenges that we had in contacting all of the focal points, but I would like to turn this onto the research communities. So the one challenge is the perception among researchers that compliance tasks like obtaining permits, registering metadata, as Josephine just said, or following the FAIR data guidelines are boring paperwork that we will do at one point to be compliant. Researchers are usually most excited about the output of their research, their manuscripts or publication. But yet these steps are integral to the Nagoya Protocol, but not only. They are essential for the ethical and reproducible science. And biobank could play a central role here as they could guarantee this traceability. Okay, let's go into a practical example. So these are the 6 steps that we defined at the beginning of the project. In order to comply with the Nagoya Protocol, but now I would like to focus on the last 2 points: the implement of governmental measures and collect permits and make them available for the BGE partners. Okay, funny story about Spain. So in our interaction with the Spanish ministry regarding the Nagoya permits, they requested that any genetic data originating from Spain includes a clear and publicly visible statement about its legal status. Nothing to do with the genetic resources, but this already, they are going into the data that are generated from the genetic resources. So to meet these requirements, we added an ethic and law disclaimer on our data portal. So now, before accessing the sequences from Spanish samples, the users of the genetic resource must acknowledge that this material and any resulting data are regulated under the Spanish law. And this notice gives compliance with the Nagoya principles on one side, by our side, but also reinforces the responsible data usage with our, as I said before, open access, let's say, framework. And given, as Melania stated before, it's not mandatory for biobanks to keep all of the permits, but I really like what DSMZ did, and it should be taken as an example. And what we did by our side is create a central repository. So there is no single universal law explicitly stating that the biobanks must keep the original permit for every sample. However, under the Nagoya Protocol and its EU implementing legislation, the users of the genetic resources are required to demonstrate the due diligence and must keep the relevant documentation for a set period of time. Usually it's 10 years, as it was said before, after the end of the use. So if a biobank is considered the user, or if all the genetic resources on behalf of the user, this is to policymaker to decide. It's not our role, but I guess the DSMZ example, it's fantastic to improve the traceability of the samples, because if all, as we did in our project, all of the project partners, so sequencing facilities, biobanks, open data repositories, and specimen providers, they know that they can retrieve the samples in one single point. And this prompt availability of the samples can help the users of the genetic resource to be compliant. And with this, I would like to thank you for your attention and mention the LIB, another branch of the LIB. So the biobank, that is the official biobank for BG, that is led, the head of the biobank is Jonas Astrid. So thank you very much again for your attention.
Thanks a lot, Christian. It was very interesting, especially the fact you pointed on the importance of compliance tasks, which are not only paperwork, but they are really relevant for the community to be followed. So thank you so much. And before we open the questions and answers session, I would like to thank everyone for having been here today. Throughout this webinar, we all have gained valuable insights into the Nagoya Protocol, its critical role in biobanks, and its implementation across Europe. And we have explored thoroughly how to navigate the protocol's provisions. We have delved into the Swiss context as an example, and learned also from real-world experiences in Swiss and pan-European projects. So as you move forward, remember that compliance, again, with the Nagoya Protocol is not just a legal requirement, but also a powerful tool for promoting fair and equitable sharing of genetic resources. And this is the most important message that we would like to bring today and to leave to the audience. I would also like to remind you that we regularly share research papers and updates from our ELSI experts via the BBMRI ERIC newsletter. And you can sign up on our website, where you will also find the ELSI knowledge base and ELSI help desk for more resources and information. And the website is bbmri.eric.eu. If you enjoyed this recording, do share it with interested friends and colleagues, and leave us a review or comment on whichever platform you tuned in via. Watch out for our next podcast episode via our BBMRI ERIC socials on X and LinkedIn. Finishing, I would like to thank you again for joining us today, and let's hope to continue fostering responsible and ethical research together. So see you next time. Thank you very much.